Assalamualaikum guys. This is a lecture for MIT 423 week 9 part 1. So on part 1 we're going to talk about immunofluorescence or IF. So what's the idea behind IF? IF is based on the nature of a molecule or substance known as fluorophore. Fluorophore is an organic molecule. IF techniques consists of labeling antibody with the fluorophore. Fluorophore is a fluorescent compound with an affinity for proteins to form a complex. So this is known as fluorophore conjugated antibody. Fluorophore will emit light under ultraviolet light. So in the reaction of fluorescence. So we can observe here UV light will make the fluorophore to emit their own fluorescent light, which can be detected later by using fluorescence microscope. So this is the basic principle on the nature of fluorophore. Fluorophore can re-emit light after its excitation by, in this case, UV light. I am using FITC as an example of fluorophore, one of the most commonly used fluorophore in medical science. But bear in mind there are a lot more types of fluorophore being used. So UV will be absorbed by the FITC, and after that FITC will emit a different light. In this case, they absorb a blue light from UV with a shorter wavelength, and then they will emit a longer wavelength light in the form of green color. And of course, the nature of antibody, based on nature of antibody, which have a very good specificity and sensitivity. So by adding an indicator system, we can find out whether the specific antibody or antigen that we are looking for is in the specimen. As an example, I put here specimen type, brain tissue. Fluorophore label antibody specific to the test antibody or antigen. So we can observe here there are three different colors which represent three different fluorophore labeled antibody, which is specific to three different antigen. The fluorophore conjugated antibody emits color when excited by UV light. So the color that being observed here is because of UV light excitation of fluorophore. The amount of color that develops is proportional to the amount of test antigen or antibody. So that means we can count the amount of antigen. For example, here we can see that the red represent the antigen for Bacterium Neisseria meningitidis in the brain tissue. So we can locate the location of antigen-antibody reaction can be observed on the specimen. This is one of the most unique features of IF, where we can locate the antigen. So this is the brain tissue. So three different fluorophore labeled antibody. So for immunofluorescence, we can have multiple color. Doesn't have to be only one. So we can for one test we can use three different specific antibody. Means we can look for three different antigen in one test, and it can be more, 5, 10. So red represent the bacterium. This is where the I mean by we can locate them. Green represent the brain cell. So this confirm that the specimen is the brain cell. And blue represent the nuclei, cell nuclei. So the background, the timeline for IF was first discovered in 1942 by Kunz, where he visualized microbial antigens. And by 1950s, IF was already being used routinely to detect viruses and other infectious agents in patient specimen and tissue culture. But on that time, they were still using non-commercial preparations of polyclonal antisera, which have significant issues with cross-reactivity. But this issue has been solved in 1980s when they introduced the use of purified monoclonal antisera. This eliminated many of the non-specific staining problems, which significant improvement in the quality and consistency of IF. And nowadays, IF remains one of the primary technologies used by diagnostic and research labs. Purpose of IF to detect antibody and antigen. Fluorescence techniques are extremely specific and sensitive, so they are very good, even compared to ELISA or EIA. IF can be used on tissue section, cultured cell lines, or individual cells, allowing the visualization of a specific antigen. And the best features of IF is that we can detect the location of the antigen or antibody in the cell or tissue. Alright guys, have a nice day. We'll continue on the part 2.